I'm going to talk through last week's PowerPoint on transport across the cell membranes. Hopefully this will make some of the more complicated areas a bit clearer. So we're going to look at the cell membrane and what it's composed of. We're going to talk about what it means to be selectively permeable, and we're going to look a little bit about diffusion and osmosis. The cell membrane consists of lipids, which are fats, and proteins, and they are arranged in a diagram like this. The lipids form a double layer and they're constantly moving, so the membrane is described as being fluid. The proteins, which you see here, vary in size and shape, and they form a patchy mosaic within the lipid bilayer. For this reason, the membrane structure is sometimes referred to as the fluid mosaic model. The proteins have a variety of functions. Some form channels, creating pores in the membrane. This allows the passage of molecules that are small enough to travel through the pores by passive means. Some proteins are partially embedded in the lipid layer, or they may lie on its surface. Some of these act as receptors for chemicals such as hormones or antibodies, and others act as enzymes for chemical reactions. Other proteins act as carrier proteins for the process of active transport of materials across the membrane. The cell membrane is described as being selectively permeable. This term refers to the property of allowing some molecules to pass from one side to another, while other molecules are unable to pass through. Smaller molecules such as water, oxygen, and carbon dioxide move freely through the cell membrane. Slightly larger ones, for example amino acids, glucose, and urea, move more slowly through it, but large ones such as proteins and starch are unable to pass through. So transport can move in and out of cells by 3 different methods. So we have diffusion, osmosis, and active transport. So diffusion and osmosis are both described as passive methods of transport. Passive transport of materials doesn't involve any energy expenditure by the organisms for the process to take place, but active transport would need some energy. So the way this works is there's this thing called a concentration gradient. In one area you'll have a high concentration of molecules such as water, and in another area you'll have a low concentration. And what happens is the movement of molecules always goes from a high concentration to a low concentration. So they'll start to move towards an area where there isn't as many of them. The definition of diffusion is the movement of molecules or ions from a region of high concentration to a region of low concentration, or down a concentration gradient. So diffusion allows substances to move within a cell from one area to another, as well as allowing materials to enter and leave a cell. Diffusion continues until the concentration of the substances is even throughout. So it needs that concentration gradient. It needs an area of high concentration and an area of low concentration in order for there to be movement. Once the concentrations are equal in the 2 areas, the process will stop. So diffusion is important to organisms in many ways. One way, for example, oxygen and glucose enter cells by diffusion for the process of aerobic respiration, and the removal of waste products such as carbon dioxide and urea from cells also happens by diffusion. At the bottom here we have our crystals of potassium penanganate, and this is what we would have shown you in school to just quickly and easily show you the process of diffusion. So when you drop a crystal of potassium penanganate in the beaker, you have a high concentration of this potassium penanganate at the bottom here, and over time the potassium penanganate will diffuse towards the area where there's a lower concentration. The colour starts to spread out through the beaker, and after about an hour the concentration within the beaker will be equal throughout, so you'll have a full purple beaker and the process will have stopped. So this slide here is just to look at that concentration gradient. So you want to see which way the sugar molecules would move. So you've got a higher concentration here and a lower concentration here. So they would move from the high concentration to the low concentration. So the high to the low. And for our salt molecules, you've got your high concentration here and your low concentration here. So they would move from the high concentration to the low concentration. So down the concentration gradient. Okay, so what substances might cells need to gain or lose? So I've already mentioned this. So you would have oxygen would diffuse into the cell where it's needed for respiration. Carbon dioxide would diffuse out of the cell as it is a waste gas. Glucose would be needed in the cell, and urea or waste would be taken out. Definition of osmosis is the movement of water molecules from a region of a high concentration to a low water concentration through a selectively permeable membrane. So it's really similar to diffusion, but it's all about water. So you need to talk about the water concentration, not just concentration. So osmosis is a special type of diffusion as it only involves the movement of water molecules. There is a difference in the water concentration between fluids inside and outside of the cell. If there is a difference in water concentration between the fluids inside and outside of the cell, the water will either enter or leave the cell, moving from the higher to the lower water concentration. If the water concentration is equal on either side of the membrane, water molecules will still move from one side to another, but in equal proportions. This is not osmosis, since osmosis requires a concentration gradient to occur. Osmosis is a very important cellular process as all cells require water to survive. Animal cells and plant cells react differently to the intake or loss of water, and it's due to the differences in their structures. So what we looked at in the last few weeks, the difference in a plant cell structure and an animal cell structure. Plant cells have a vacuole for water storage, and any water gain or loss from a cell affects the size of that vacuole. And this has an overall effect on the rest of the cell, as shown in these diagrams. So a plant cell in a solution which has an equal concentration to the solution in the vacuole will experience no net gain or loss of water, so the cell will remain unchanged. The one we have in the middle here. So we've got the cell we're comparing it to, and then when there's an equal water concentration, the size of this vacuole remains the same and overall the cell remains the same. However, if the solution surrounding the plant cell has a water concentration greater than that cell, this one here, water passes into the cell from the surroundings, causing the vacuole to swell and press outwards towards the cytoplasm. So here you can see that this vacuole has increased in size. This in turn causes the cytoplasm to push against the cell membrane and the cell wall. So here you can see that the cytoplasm is starting to push outwards on this cell wall and cell membrane. The cell wall is fairly rigid though, and it's strong enough to resist this pressure, so it does prevent the cell from bursting. And how we describe this cell here is turgid. If the plant cells put in a solution that has a lower water concentration than the outside, then the water starts to pass out of the cell into the surroundings, so it causes the vacuole to shrink. So you see here the vacuole is a lot smaller in size, and this causes the cytoplasm to pull inwards, and it drags the cell membrane inwards too, but the cell wall will remain in place. So you start to get these gaps developing between the membrane and the cell wall, and as the cell wall is fully permeable, the surrounding solution flows in to fill this gap. The way we describe this cell is plasmolysed. So animal cells react differently to plant cells in terms of water gain and loss, and this is due to the fact they don't have that cell wall and they don't have a vacuole. So an animal cell in a solution which has an equal concentration to the solution in the cell will experience no net gain or loss of water, and it will remain unchanged. Okay, so equal water concentration, and it's exactly the same. If the solution surrounding the animal cell has a water concentration greater than the cell, the water passes into the cell from the surroundings, and this causes the cell to swell up. As there's no cell wall, the cell membrane continues to stretch and eventually the cell bursts. On the other hand, if the solution surrounding the cell has a water concentration lower than that in the cell, water passes out of the cell to its surroundings and causes the cell to shrink and shrivel up. As red blood cells are surrounded by a solution, blood plasma, it is important that the body has mechanisms in place to prevent the plasma becoming too concentrated or dilute, as it can cause your red blood cells to shrink or burst. The definition of active transport is the movement of molecules and ions across the cell membrane against a concentration gradient. So in diffusion and osmosis, you had molecules moving from a high concentration to a low concentration, so it was going down the concentration gradient. But in active transport, they go the opposite way. So they're going from a low concentration to a high concentration. So you have to think about it as you're walking up a hill or walking up stairs, that takes extra energy, that needs energy to happen. So unlike diffusion and osmosis, molecules or ions do not move by themselves in active transport. Instead, they are moved by carrier proteins, which are a component of the membranes that we talked about earlier. And this process requires energy because molecules are being moved against that concentration gradient. So they're going from a low to a high concentration. The key things not to forget in this subtopic is diffusion and osmosis are two types of passive transport. So neither of those processes require energy expenditure by the organism. And then active transport goes against the concentration gradient, so that requires energy expenditure by the organism.